in first, I thanks to Andrea for the invitation. For me, it's a big opportunity to show our work in the SLAMTB. Well, I am a professor of University of Santa Cruz do Sul in a graduate program in health promotion. Andrea Valim is my colleague and is professor too in the same post-graduation program. And today she can't stay here with us. Well, Santa Cruz do Sul is the city located at Rio Grande do Sul in South Brazil, in the center of the state. It's far from the capital, around 150 kilometers. Here is Rio Grande. Unisc has 27 years and is a community university. We have 5 campuses in Rio Grande do Sul state, 73 undergraduate programs, 9 postgraduate programs with 6 PhD programs and around 7,000 students. We have a hospital, university hospital called Hospital Santa Cruz, and this hospital has 232 beds, 5 medical houses, and 1 multidisciplinary house. Our labs are located inside the university campus. We have a biotechnology research training center associated to technological and scientific park, genetics and biotechnology laboratory, and laboratory of cells cultivation. And we have a partner, Regional Central Laboratory, to realize tuberculosis culture tests. Techno-Unisc, our scientific and technological park, promotes the investigation between the triple helix agents, universities, society, and governmental institutions in an environment of entrepreneurship, research, and technological innovation. Hence, aiming at the sustainable competitive advantage of the organizations and their effective participation in the economic and social development of the region. Our post-graduation program is in the promotion health area. This is a program with 10 years. We have 12 researchers, 6 staff members, 12 master students per year, and 13 PhD students. This is our first group. And we have a journal related to the post-graduation program and to university hospital. This journal is one of the vehicles for the dissemination of experiences and scientific studies related to the issues of surveillance and the control of infection related to the notifiable disease, infectious disease, occupational health, and related areas. Now I talk about our research lines in tuberculosis that our group done here in Santa Cruz do Sul. Tuberculosis in prison, health surveillance, molecular epidemiology, TB, HIV, and latent tuberculosis are the lines that we study here. But actually, due to the pandemic, we are studying COVID-19 in the diagnosis by PCR, and we are participating the EpiCovid study from Federal University of Pelotas. But today I will talk about specifically about tuberculosis in prisons, the history of the studies that we are doing here. In Brazil, the prison population increased around 700% in 26 years. But the infrastructure and vacancies don't accompany this increasing. In this line, in this green line, is the total prisoners in 2020. The red line is the deficit of vacancies, and the blue line is the total of vacancies. But we can see that this difference, and we can see that the overcrowded is a big problem in the Brazilian prison system. Unhealthy housing, difficult to access health services, high endemicity of TB inside the prison, drug abuse, overcrowded, and frequent time of incarceration are the determining factors for TB load in prison. TB in prison here in Brazil is around 28 times higher than in a general population. Then our first study here in Santa Cruz do Sul, in our first study, we studied the spatial distribution and epidemiological characteristics of TB cases in the urban area of Santa Cruz do Sul. And in this study, we can see that the spatial distribution of cases was concentrated in southern urban area, where the socioeconomic status is lower. Here, where is appointed. And this place specifically is inside and around the prison, the regional prison here in Santa Cruz do Sul. During all period studied, the same place were identified with higher prevalence. Then we start a new project inside the prison. We studied prevalence of respiratory symptoms and active tuberculosis in this prison, prevalence of HCV, and doing these studies and being aware of the health reality inside the prison, we have started to ask, how is the health team working related to tuberculosis control inside the prisons? Then, here in Brazil, we have a public policy of health for prisoners called PNAISP, Política Nacional de Atenção Integral à Saúde das Pessoas Privadas de Liberdade no Sistema Prisional. And this policy established the prison primary health care team inside the prison. And these teams, the functions of these teams are health promotion, infectious disease prevention, vaccines, oral and woman health, acquisition and transfer of medicines from the basic pharmacy, and other activities. These teams are composed by nurses, dentists, medical auxiliaries. In Rio Grande do Sul state, we have 26 primary health care teams distributed in 20 cities. These teams cover around 70% of the prison population, the attendance of this population. Well, then we started to study these teams and the actions in surveillance, in tuberculosis surveillance. And two studies that we done, we identified lack of knowledge of the team about health networks, teamwork, monitoring, and surveillance tools. These teams do not receive specific training to work inside this prison system. And this is a big problem to the diagnosis of tuberculosis and other infectious disease. Then, in this sense, in the next step, our group developed a training protocol for this team addressing the following themes: health networks, territory surveillance, organization of care for acute events and chronic conditions in primary health care, pharmaceutical care, teamwork, evaluation, and monitoring. And this protocol, this training, provided an exchange of knowledge, ideas, and attitudes to proposing different ways of developing the team's individual and collective work. The knowledge derived from the exchange of experience and the acquisition of knowledge is transformed into opportunities for changing habits and empowering workers in their workplaces. Related to molecular epidemiology, our first study in prisons were here in Santa Cruz do Sul, and we published this work in 2016 about recent transmission of drug-resistant tuberculosis in a prison, where we conducted a cross-sectional retrospective study characterized by epidemiological and molecular epidemiology. And in this study, we analyzed by Miro VNTR 53 isolates, and we identified around 30% of these in clusters, and 85% of the resistant isolates are inside of a unique cluster. And the new project that we elaborated these years by many collaborators from different institutions, we submitted to the grant of Health Ministry, but unfortunately we was not approved. But this project is divided in 5 axes. Axis 1, molecular epidemiology, surveillance and monitoring. Axis 2, Axis 3, information technology, diagnosis, and social control. Axis 1 and Axis 2, we are studying inside of the projects of master and PhD projects. Now I will talk about the preliminary results about genetic diversity of Mycobacterium tuberculosis isolates circulating in prisons of South Brazil. Until now, we analyzed by Miro VNTR 236 isolates from patients of 15 prison units. 69 isolates was in clusters. In 9 clusters, prisoners were in the same prison at the time of diagnosis. And in 10 clusters, prisoners, they were not all the same penal institution at the time of diagnosis. 98% is male, around 53% had 18 to 30 years old, 45% have 4 to 8 years of study, 18% has co-infection TB at A grade B, and 63% of the patients have cured their treatment. In relation to resistant profile, we identify around 12% of the isolates resistant and around 4% in MDR. We studied also the phylogeny and identified around 50% of the isolates inside the lung family and around 40% inside the Harlem family. And to finish this presentation, I would like to talk about social impact of this project in our community, because we work not only with research, we work with research, extension, and teaching. Here at Unisc, these three things are very enclosed, very linked, because we are a community university. Then it's so necessary that we organize some events, seminaries, workshops, congress, mostras, to take this information, the scientific information, to our community. Then I thank you, and this is our group, our professors, students, and thank you very much.